Hello, everybody. Welcome you at the fourth edition of the Renal Pathology Conference. Ladies and gentlemen, dear participants, we are delighted to welcome you on behalf of the organizing committee. This edition takes place in Warsaw. The meeting is virtual. We do hope that it will be exciting and fruitful. We are happy to announce that the conference has aroused wide interest among pathologists and nephrologists around the world, since we have more than 500 attendees from 23 countries. Let me introduce Professor Gaciąg, Director of the Medical University of Warsaw.
Dear guests, welcome to the fourth International Renal Pathology Conference. My name is Zbigniew Gaciąg. I am the president of the Medical University of Warsaw. As a physician, for many years I was directly involved in the renal transplantation program, so I still consider myself a member of kidney community. This is an exceptional meeting, which is very special because this is the meeting which goes directly to you, to your homes, to your offices. This is the meeting with 32 sessions, the sessions which are chaired and has speakers from all around the world, the experts in many fields, not only related to renal pathology, but to different areas of medicine and science. This is the meeting that offers an opportunity both for scientific exchange as well as educational activities. So therefore, organizing committee and scientific committees should be congratulated, and I would like to pass my congrats to Agnieszka Perkowska, who is chairing this committee and made a tremendous effort for this event to take place. So therefore, officially, I announce that the fourth International Renal Pathology Conference is open.
Thank you so much, dear Professor. We are also very pleased to welcome Professor Michael Mengel, the President of Renal Pathology Society. Michael, we are ready for your opening speech.
Yeah, thank you, Agnieszka. I hope everybody can hear me. Good morning from my end of the world. I'm in Western Canada. It is 4:30 a.m. But this is the beauty of a virtual conference around the world, that we all can participate and share knowledge from everywhere we are at the moment and whatever we do. So I really would like to echo the words, the kind words of the President of the Medical University of Warsaw, and welcome you all to the fourth International Renal Pathology Conference. And I look forward to a very exciting event with a virtual format, but nevertheless more engaging and exciting to share the state-of-the-art knowledge in renal pathology across the world. We have a wonderful program, and the organizing committee did an exceptional job in putting this together and transitioning it from a first in-person meeting, but then due to the pandemic, to a virtual meeting in a very efficient and exceptional manner. So with that and no further delays, I also would like to welcome everybody and look forward to a very exciting event. Thank you, Agnieszka.
Thank you so much. We do very much count on your active participation during the conference. So please send us questions and comments concerning the lectures through the chat box. They will be addressed at the time of questions and answers sessions. In case we do not have enough time for the questions asked through the chat box, I promise that these questions will be answered by the lecturers afterwards. Now let us move to the first session. Please welcome our first two moderators, Professor Magdalena Durlik and Professor Andrzej Oko.
Professor Magdalena Durlik is the head of Department of Transplantation Medicine, Nephrology and Internal Diseases at the Medical University of Warsaw. Professor Magdalena Durlik is also the past president of the Polish Transplantation Society, honorary member, and the past president of the Polish Society of Nephrology.
Professor Andrzej Oko is the head of the Department of Nephrology, Transplantology, and Internal Medicine at the Poznań University of Medical Sciences. Professor Oko is currently the president of Polish Society of Nephrology. Professor Durlik, Professor Oko, the virtual floor is yours.
Thank you very much. Good afternoon, ladies and gentlemen. It is my pleasure to welcome you to the International Conference of Renal Pathology. And let me welcome the first speaker, Professor Tobias Huber from Germany. Professor Tobias Huber is head of the Center of Internal Medicine of University of Hamburg, and his lecture is entitled COVID-19 and the kidney. So hot topic. Professor Huber, the floor is yours.
Dear colleagues, first of all, I would like to thank the organizers and the scientific program committee for having put together such a fascinating meeting. I would also like to applaud them for having so swiftly changed the meeting from an attendance meeting in spring to a now virtual meeting in fall. Now, with changing this meeting, I also switched my title, and I'm now focusing on the very actual theme of COVID-19 mediated kidney disease. Now, very early already in this COVID-19 pandemia, it was recognized that many people suffered from kidney injury, from kidney failure, and from the large demand of dialysis. So as stated here in the New York Times, there was no shortage of ventilators at the time, but rather of dialysis machines. And then when the first cohorts got analyzed, and this is an analysis out of the United States again, it became very clear that a high percentage, up to 40% of the people having been admitted to the hospitals suffered from acute kidney injury, and as much as 30% of these patients suffered from stage 3 acute kidney injury. And the occurrence of acute kidney injury, particularly severe acute kidney injury, was associated with the outcome of the patients and mortality and morbidity rates. This shows from the first COVID wave, the analysis of 10,000 patients that has been in hospital in Germany. And again, as you can see here, the prognosis worsened dramatically if people were on ventilators, and it got worse if patients were on ventilators plus dialysis machines, up to a death rate of over 70%. But COVID-19 was also a threat to already chronically diseased kidney patients and dialysis patients, and we organized for the German Society of Nephrology from Hamburg and COVID-19 dialysis registry in Germany. And here you can see our data that the incidence was around 2% of patients being positively tested for SARS-CoV-2. And the mortality among this cohort was up to 20%, confirming that COVID-19 is not only causing acute kidney injury and severe acute kidney injury with dialysis, but also it means major threat to our dialysis population. Now, all this got us very curious to understand more on SARS-CoV-2 actions outside the lung, and we ask ourselves whether SARS-CoV-2 might infect other organs than the respiratory systems. Now, with a large team at the UKE in Hamburg, including also the pathologists, led by Thorsten Wiech, our legal medicine department performing all the autopsies led by Klaus Püschel, and also our microbiology department led by Martin Epfelbacher. We had access to one of the, I guess, worldwide largest autopsy cohorts to analyze tissue samples and to correlate it to the clinical data. And this was our first observation and first publication where we tested throughout the body whether SARS-CoV-2 can be found in other organs than the respiratory system. The study was led by Victor Puelis, who is an assistant professor in our department. Now, what we found is when we analyzed 27 patients that died by COVID-19, that all these patients had a high number of comorbidities. The average was 3.3 severe comorbidities like cardiovascular disease, like chronic kidney disease, like liver disease, like cancer, or other severe organ diseases. Next, when looking at these patients, we realized then that the number of comorbidities correlated to the finding and detection of the virus in other organs. The more comorbidities the patients had, in the more organs we found SARS-CoV-2 detected by PCR analysis. And interesting, this was, there was a particularly clear association in the kidney and kidney tropism. The more comorbidities, the more often we found SARS-CoV-2 in the kidney. Now this compares different organs, and as you can see that when we quantified the RNA copy number of SARS-CoV-2 per cell in different organs, that not only in lung and pharynx SARS-CoV-2 was found, but in multiple other organs SARS-CoV-2 was present. And among these other organs, the kidneys were the organs being mostly infected and at relatively high rates, followed by liver, heart, and much less in brain and blood. Now, when asking ourselves at the early time point of the first wave, what might be the reason that we find SARS-CoV-2 so commonly and also in high copy numbers in the kidney, we questioned ourselves whether the receptors are present for SARS-CoV-2 in the kidney, and these receptors, namely ACE2, have been known for a time, and they are also receptors for SARS-CoV-1. In addition, membrane-bound enzymes are important, cleaving ACE2 and facilitating the process of virus absorption. As you can see, by single-cell analysis from fetal as well as adult kidney, we could find an abundance of ACE2 as well as the facilitator genes. Now, this time it was very important to apply different techniques to detect the virus, to make sure that there is true virus in the organ. So the first detection line was done by PCR from tissue. One expert was establishing in situ hybridizations to directly detect the viral RNA in the tissue. And as you can appreciate here, that you can see it in the lung, but it can also be detected in glomerular tissue as well as in tubular tissue. Next, we established immunofluorescence-based detection methods of SARS-CoV-2 protein. And again, you can see this positive control in the lung, but we could detect it now throughout epithelial compartments in the kidney, including glomerular structures, likely endothelial cells and podocytes, but also prominently in tubular structures. Now, when we next looked closer at different and fractionated kidney tissues to look at different compartments, it turned out that we detected quite a bit of virus in glomerular tissue as well as in tubular tissue. Now, this might have been overlaid by the fact that the tubular tissue was much more autolytic than the glomerular tissue, which might lead to an overestimation of glomerular tissue. But at least there is clear detection in glomerular tissue by all three methods: PCR, in situ hybridization, and direct protein detection. And this might very well relate to the findings that have been reported by several groups and case reports that few, or in rare cases, COVID-19 patients develop a collapsing FSGS. Now, let me summarize the first part of our observations. So basically, we found and redefined a respiratory SARS-CoV-2 virus to a multi-organ virus with particular renal tropism. So SARS-CoV-2 infects multiple organs beyond the respiratory tract. The kidneys are very prominently affected, and comorbidities play an important role in the viral spreading throughout the body. And this might be one explanation to the broad range of clinical symptoms that we observe in these patients. While surely their systemic inflammation, cytokine release, hemodynamic factors, many factors play a role for this broad spectrum of phenotypes, we have to take in consideration that some of these clinical symptoms might also be caused by direct viral infection of certain organs, particularly the kidney. Now, the next question that we try to address is whether the SARS-CoV-2 tropism in the kidney associates with organ outcome. And for this, at the time, again, we did one of the largest autopsy studies, looking at 63 autopsy cases. And this study was led by Fabian Braun as the first author, and also Victor Puelles, who played an important role, and many, many others. Again, you know, all the teams of microbiology, pathology, legal medicine, and of course, from our nephrology team and our departmental team were involved. This shows an overview of over 63 autopsy cases and their clinical characterization. And as before, we witnessed lots of comorbidities in these patients that died by COVID-19. Again, more than 3 comorbidities on average. And again, like in our first cohort, we detected in 60% of these patients SARS-CoV-2 virus in the kidney. And the presence of SARS-CoV-2 in the kidney was associated with higher age, with the number of comorbidity, and slightly with male sex. Now, what does it mean if SARS-CoV-2 can be found in the kidney? The first finding, which was quite unexpected, was that if we find SARS-CoV-2 in the kidney, the survival of these patients from the initial detection of SARS-CoV-2 in the lung to the time point of death was significantly shorter. So viral spreading, including the kidney, was a predictor for shorter survival time. Now, most importantly, viral detection and renal tropism of SARS-CoV-2 was associated with a significant higher number of acute kidney injury, and particularly also severe acute kidney injury. Well, then the question is, if there's a clear correlation of SARS-CoV-2 renal tropism to outcome, whether there is not just an infection and a detection of SARS-CoV-2, but also an active replication of SARS-CoV-2 in the kidney. And for this, we used the supernatant of kidney pieces from patients that died by COVID-19, and then we took the supernatant soup and infected cells and measured whether it could infect cells and whether the virus is replicating in these cells, and we found that the virus was highly active and replicated by measured here 1,000-fold increase within 48 hours. So this kind of is like a Koch-Lesch's postulate. So we can take and transfer virus from kidney tissue to other cells and organs where it replicates as proof of concept that SARS-CoV-2 is replicating in the kidney. And this could be further substantiated when we used non-human primate tubular cells that we infected with SARS-CoV-2 and stained for replicating markers and could see a strong replication in these cells. Let me summarize this part of the presentation. So we confirmed again in a second and at the time largest autopsy study the renal tropism. And we could show that renal tropism associates with an earlier and premature death, and that renal tropism associates with acute kidney injury. And in addition, we could show that SARS-CoV-2 actively replicates in the kidney, indicating that COVID-19 nephropathy might be a direct result of virus action. Let me summarize this part using one schematic that we very recently summarized our data. And we think that major factors driving kidney failure are, of course, hemodynamics, microthromboembolisms, cytokines, and inflammatory status. But our data also suggests that direct viral entry and infection of kidney epithelial cells contribute to the outcome and to the clinical phenotype. And along this side, there was also an interesting paper from Belgium showing that early on there is tubular functional defects as shown by low molecular weight proteinuria, uricosuriya, phosphaturia, and others that could likely be an effect also of a functional disturbance by a viral tubular infection. Now, when we talk about cytokines, it's of course important to get a better understanding of the cytokines and the inflammatory reaction, and lots of papers have been published already. I just want to show one slide also of our departmental team led by Christian Krebs and Ulf Panzer, who are studying immunology in the kidney usually, but here they looked at broncho lavage fluid and did a very deep single cell analysis to detect that there are a certain type of tissue resident T cells that are being activated and that are probably releasing pro-fibrotic and pro-inflammatory factors, driving in this case inflammation in the lung. So I think at the moment we are learning a lot, not only about the innate, but also the acquired immune response. And this is now linking it also to tissue resident compartments and acquired immunity. Now let me come to a bit more outlook and the clinical application and relevance of these findings. Now, in a hypothesis paper, which was led by Oliver Gross and it was done together with us, we speculated that because of our data showing that SARS-CoV-2 has a strong renal tropism, that maybe very early urinary changes could be a predictor for later outcome. So we suggested that every patient being admitted to a hospital should be analyzed, a very simple and cheap analysis of the urine just by dipstick and also albumin and protein to creatinine ratios. Now, based on our hypothesis, which was also published in May, we then followed up a clinical cohort analysis, again led by Oliver Gross together with our department in Hamburg and also the University Medical Center in Aachen and the hospital in Cologne, Meerheim. And then basically group patients at admission to normal or altered urinary status, and also included two serum parameters that we measured that we had been observed earlier that they correlate with a bad outcome, which was AT3 and serum albumin. And based on these simple parameters, just the urine parameters, plus the serum parameters, we generated traffic light systems in low, intermediate, and high risk. And very interestingly, just on these very simple tests at hospital admissions, they correlated with the later outcome, meaning the percentage of patients being admitted to ICU or dying. If they had no urinary abnormalities at the beginning, this rate was much lower than if they had urinary abnormalities and in addition, changes in serum albumin or serum AT3. And even without taking into account these two serum parameters that we included in our analysis tool, again, there was a big difference just by the presence or absence of urinary abnormalities, showing that early urinary abnormalities are a very interesting predictor of outcome. Now, let me summarize. In summary, we were showing that there's a renal tropism of SARS-CoV-2 for the kidney in concert with the inflammatory status, cytokines, and hemodynamic factors. This might contribute to the high prevalence of kidney diseases. And again, as also shown in this News and Views accompanying one of our articles, SARS-CoV-2 is a pleiotropic, multitropic virus affecting different organs, particularly the kidney, that then can lead to specific kidney disease eventually, like collapsing FSGS, but also promote and facilitate AKI. With that, I would like to thank all the people that contributed. Again, this was a true collaborative work in Hamburg, and it would have never been possible with all the autopsy work by the legal medicine, but also pathology and microbiology. And from our team, quite a few people have been involved working day and night during this crisis. And most importantly, Victor Boeles, Fabian Braun, Maya Lindenmeyer, Nicola Wanner, while all others also helped. With that, I would like to thank you for listening wherever you are, and I'm happy to respond to questions which hopefully appear now via the chat function. And I would also like to thank again the organizers. Thank you for listening.
Thank you very much, Professor Huber, for your very interesting lecture. Discussion will be held at the end of the second lecture, so thank you. Please wait for discussion.
Now I have a great pleasure to introduce Professor Andrzej Więcek, past president of IRA-EDTA. In the past, he was also president of Polish Society of Nephrology. And since several years, he is head of Department of Nephrology, Transplantology and Internal Medicine at Silesian University of Medicine. Please, Professor Więcek, this virtual floor is yours now.
Good afternoon. My name is Andrzej Więcek, and I'm very pleased to participate in this 4th International Renal Pathology Conference. And I would like to thank Professor Perkowska-Ptasińska for her kind invitation to this very prestigious conference. The topic of my lecture today is mechanism of proteinuria. And we all are aware that proteinuria is one of the most important symptoms of kidney diseases. And it is reported also that proteinuria is one of the most important risk factors for cardiovascular diseases, stroke, and progression of chronic kidney disease. However, the etiology and also pathogenesis and clinical significance of proteinuria is not fully understood yet. So during this the coming 20 minutes, I will give you an overview. What are the current concepts of proteinuria? I have nothing to declare concerning the topic of my lecture. So I would like to start with these two statements that proteinuria may result from two principal mechanisms. First is excessive permeability of the glomerular barrier. And second, proteinuria may also result from impaired reabsorption of protein in the proximal tubule. It's also possible that is a combination of the two above mechanisms. And I would like to refer you to this very important, and I have to say, the seminal paper published many years ago by Giuseppe Amico and Claudio Bazzi from Milano, Italy, where they very elegantly describe the pathogenesis of proteinuria. And at that time, it was very novel concept and novel description of proteinuria. So first, they gave the picture with the physiological conditions where even there is a slow permeability of albumin or low molecular weight proteins through the filtration barrier. But this small amount of these low molecular proteins are entirely absorbed by the tubular, proximal tubular cells. So there is no proteinuria at all. In case that there is a larger amount of low molecular weight proteins or large amount of albumin filtered through this filtration barrier, and this reabsorption of the proximal tubule is already saturated, then more albumin and low molecular protein is present in the urine, and then usually it's also on a very few amount of the high molecular weight proteins, like for example immunoglobulins, that then we say that we have a selective proteinuria. And then when this damage of the filtration barrier is greater, also a larger amount of high molecular proteins are filtrated, and of course there is already the transport through the proximal tubular cells is saturated, and then in the urine there is both low molecular proteins and the high molecular proteins, and we say that we have a non-selective proteinuria. And when the progression of the disease is present and there is a severe glomerular damage and also tubular damage, then there is a massive proteinuria and also damage of the interstitium because of the inflammation and fibrosis processes. So and we can distinguish when we have only the tubular damage, then we can say that we have the tubular proteinuria when the proximal tubular cells are unable to absorb these proteins filtrated in the glomerulus, and when there is a massive damage of filtration barrier, then we say that we have a glomerular proteinuria. And And we can distinguish this because usually the tubular proteinuria is low. As you can see here, the albumin to creatinine ratio in the tubular proteinuria is below 0.4. And in the glomerular proteinuria, this albumin to creatinine ratio is higher. And there are several diseases mentioned here. And usually the tubular proteinuria is present in tubular interstitial nephritis or acute tubular necrosis, or in and for example, also in myeloma cast nephropathy. But in the glomerular diseases, and there are many different types of glomerular diseases, the proteinuria is much, much higher. And you see here different types of glomerular diseases with much higher proteinuria. So it's just an indication how to distinguish the tubular proteinuria and the glomerular proteinuria, because the tubular proteinuria is usually very, very small. What is the function of proximal tubular cells in their absorption of the proteins which are filtrated in the glomerulus? There's a very complicated mechanism usually involved. There are two transporters are involved, megalin and kubulin. The megalin, as you can see here, transports this low molecular weight proteins and also albumin into the proximal tubular cells. And then this low molecular weight proteins are biodegraded in lysosomes, and there is no protein in the urine. Of course, this mechanism is not sufficiently enough. When the proteinuria, the glomerular proteinuria is higher, then this mechanism is saturated and the protein is present in the urine. And what are the consequences of this transport of albumin or low molecular weight proteins into the interstitial space through the proximal tubular cells? The consequences are inflammation and also fibrosis of this interstitial part of the kidney, and of course this also may affect the proximal tubular cells, which additionally may decrease this capability of absorption of the low molecular proteins which are present in this lumen of proximal tubules. So you see here when the protein is absorbed by the tubular cells, and this protein is already in the interstitium, it creates a lot of reactions, and many cells are involved, T lymphocytes, macrophage, and the fibroblasts, myofibroblasts, which may generate a lot of cytokines and pro-inflammatory factors, and also pro-fibrotic factors, which in consequence may lead to the interstitial fibrosis and inflammation. So, and it's well known that proteinuria, and especially the higher proteinuria, is a risk factor for the faster progression of chronic kidney disease. And one of the reasons is that this protein is absorbed by the tubular cells and creates this pro-inflammatory and pro-fibrotic reactions, which I described before. This was the role of tubular cells in the proteinuria and the consequences of absorption by the tubular cells proteins which are in the lumen of the proximal tubules. The most important, however, role in proteinuria is the filtration barrier and the damage of filtration barrier. And the podocytes, these are the most important cells which participate in the normal activity of filtration barrier, and also damage of podocytes may lead to massive proteinuria. And this is a very nicely demonstrated on this picture, where we have a model of glomerular filtration barrier in the normal conditions, where you see the food processes of podocytes. We have a quite normal glomerular basement membrane and also endothelial cells. And also in this case, albumin, which is presented here, is not able to cross this filtration barrier because these podocytes food processes of podocytes are well attached to the glomerular basement membrane, and this is only permeable for water. However, in the disease situation, when you see here the food processes are destroyed, and this is not a very strong connection between these food processes where the slit diaphragm is located. This is much brighter, this area. And also, as you see here, the structure of glomerular basement membrane is different than here. So then this filtration barrier is damaged, and albumin, and also in later stages also globulins are able to cross this filtration barrier and are present in the urine. So the first description of the so-called slit diaphragm was published many years ago in 1974 using the electron microscope. The authors, they observes so-called zipper-like morphologic structure, and they concluded that it might be important also for the filtration. And then the next step was a very important step, was to discover nephrin with some immunostaining methods, and you see between the food processes just the location of nephrin. And then Carl Tryggvason from Stockholm, Sweden, he was one of the first to very precisely describe the structure of the slit diaphragm, and he proposed that nephrin is the main factor which is located in the slit diaphragm. And as you can see here, there are the food processes from two podocytes, and in between there is an overlap of this nephrins from one and from the second podocyte, and in between is this slit diaphragm. He also proposed that in between maybe also some pores, which can also be important in the pathogenesis of proteinuria. As you can see here how it works, this is a slit diaphragm between the food processes of podocytes, and there are endothelial cells and the glomerular basement membrane in between. And when this structure is complete is normal, this is not capable, there is not permeable for albumin or for protein. Now this knowledge is much bigger, and we know that this is not only nephrin in this slit diaphragm, but also other proteins and other factors. So the slit diaphragm is much more complicated, and also the food process from podocyte is also much the composition, the components of the food process is much more complicated, and so the structure of this system is, as you can see, very complicated. And this is not only to know that the slit diaphragm is important, but also the structure and the so-called cytoskeleton of the food processes is important, because when they are flat, when they are not well attached to the glomerular basement membrane, this structure does not work correctly and is also permeable for protein. And in normal physiological conditions, there is a very strong interaction between podocytes, the slit diaphragm, and this is, we say that there is dynamic interaction, because as you see here is a lot of factors involved in this very complicated structure between food processes and slit diaphragm, and usually this is not permeable for albumin. However, in different conditions, this dynamic interaction between food processes and slit diaphragm is broken, is damaged. And of course, there are different mechanisms how this dynamic interaction is damaged. Is different in diabetic nephropathy, is different in minimal change disease, where mostly is a problem is nephrin, sometimes it's a mutated nephrin. The same is true for the focal segmental glomerulosclerosis. However, membranous nephropathy is also different mechanisms. Usually it's a PLA2 receptors are involved. So these are different mechanisms which destroy this dynamic interaction between food processes and the slit diaphragm, leading to proteinuria. Finally, I would like to say that there are some other mechanisms which may participate in proteinuria, or at least in albuminuria. One of these mechanisms is transcytosis, and we can add transcytosis as the third potential mechanism of proteinuria. And we have to know that transcytosis may occur in all these cells which are present in tubular proximal tubular cells, in podocytes, or in endothelial cells. Through these cells, this transcytosis may occur, and of course, this transcytosis may occur in both directions. And many different factors are involved in this transcytosis: angiotensin II, vascular endothelial growth factor, and so on. And for example, this is the transcytosis of albumin in the endothelial cells, where the albumin is transported from the blood to the urine. And as you can see, the albumin is transported in so-called small vesicles, which are created by caveolins, as you can see here, and also many other factors participate in this creation of these vesicles from the caveolins. And on the other side, there is exocytosis, where the albumin is secreted to the primary urine. And this model, for example, is also important in diabetic patients, where high glucose may accelerate this endocytosis through endothelial cells. And there are some ideas that in the early stages of diabetic nephropathy, the albuminuria and proteinuria is mostly due to this mechanism. So there are the three different mechanisms of proteinuria: damage of the slit diaphragm, damage of food processes, and also damage of glomerular basement membrane, and finally the transcytosis. So it's presented also here that albumin is, by the damage of slit diaphragm, is permeable to the primary urine. The proximal tubules may absorb the protein, especially albumin, by the megalin-kubulin pathway, but the system is only partially effective. And also the transcytosis, which in this situation may go from the lumen to the blood, but also from the endothelial cells, as in diabetic patients, for example, goes from endothelial cells to the primary urine. Finally, I would like to say a few words about glycocalyx as also one of the mechanisms of proteinuria. And usually in the physiological conditions, the glycocalyx protect the permeability of albumin and protein into the Bowman capsule. It's the endothelial cells, as is a glomerular basement membrane, and podocytes, and this is a physiological condition. Usually, this glycocalyx is negatively charged, and also albumin is negatively charged. Therefore, albumin is pushed out from the endothelial cells. It's not possible to go for albumin into the interstitial space. However, when this glycocalyx is destroyed, there's a calcium, which is, as you see here, positive, may increase this permeable of albumin between these endothelial cells, and may participate in albuminuria. So destroy of glycocalyx, which is the negatively charged, increases permeable of albumin through the endothelial cells. And this is also very important in diabetic patients. As you see here, high glucose through different mechanisms destroy glycocalyx and increase permeability of albumin. And the final word is some genetic background of proteinuria, and we know that there's several genetically determined diseases for podocytes, the red color, glomerular basement membrane, the blue, and endothelial cells, the green color. And these are here many genes which were already described, and when there are some mutations in these genes, this may lead to the severe proteinuria. And with severe proteinuria, of course, is one of the, as I mentioned already, one of the risk factor of the glomerulosclerosis. So this is the conclusion that when we have increased glomerular capillary pressure because of the loss of nephron, we have podocyte dysfunction, increased glomerular permeability for macromolecules, for protein, for example, increased filtration of plasma protein, excessive tubular reabsorption, but this is a limited process and is quite often is saturated with this protein and is not efficient anymore. And then there's a release of vasoactive inflammatory proteins in the interstitial tissue, which leads to further scarring and GFR loss. So in conclusion, proteinuria is one of the most important symptoms of kidney disease and is reported to be the most important risk factor for cardiovascular disease, stroke, and progression. Etiology is not clear. I presented to you some mechanisms of proteinuria, and the most important is this glomerular proteinuria, which is associated with pathological damage of the glomerular filtration barrier. We have also tubular proteinuria, which is caused by the damage of proximal tubular cells. And albuminuria, proteinuria may be also caused by other mechanisms like, as I mentioned, tubular dysfunction, but also transcytosis and loss of glycocalyx on the endothelial cell surface. Thank you very much for your attention.
Thank you very much, Professor Więcek, for this exciting presentation.
Now we have time for a few questions. Yes?
Yes. Yes, of course. Please, please, Professor Durlik.
May I have a question to Professor Huber concerning SARS-CoV-2 infection? My question is, what about detection of the virus in the urine? Because similar to BK polyoma nephropathy, we can detect virus in the urine. It seems simple diagnosis. What about virus, SARS-CoV-2 virus and detection in the urine?
Yeah, this is, thank you very much. This is, of course, a good question. And people have been trying to detect a virus in the urine, and some could and some couldn't. This depends on the detection methods. And then we don't know, of course, where the virus is coming from. You know, this could be virus that circulated in the blood and it has been filtrated, or it could be virus coming from tubular cells that contain virus and have been shedded into the urine. So then this is, you know, always there's not 100% clear where the virus is coming. So this is why I think it doesn't play a big role right now for diagnostics.
Thank you.
I have a question to Professor Więcek. As you know, in minimal change disease, there is sometimes heavy proteinuria, and we don't see evident kidney damage for several years in spite of this heavy proteinuria. In IgA nephropathy, proteinuria above 2 grams per day is a marker of worse prognosis. In contrast, in membranous nephropathy, proteinuria below 4 grams per day is a marker of good prognosis. Could you explain this discrepancy between these pathological conditions?
Of course, there is just a place for the speculation. There is no single answer to your question. Of course, there is a different pathogenesis of these diseases. So this is not, for example, in minimal change disease, this is just mostly the problem is in nephrin and the mutation of nephrin, or some just very simple, very small damage in this filtration barrier. In the IgA nephropathy or in the membranous nephropathy, you have also some other immunological additional factors which may, in my opinion, also lead to the damage of, for example, the tubular cells. And I think that the composition of the damage of the filtration barrier and tubular cells may increase the risk of the fibrosis or the local interstitial inflammation. So the simple answer to your question is that the proteinuria in these conditions which you mentioned is caused by different background, and this additional immunological status may also influence the prognosis.
Thank you very much. I think we can say that podocytes are main most important cells in the glomerulus. Thank you very much.
And we have also several questions from other participants. So the questions coming from Professor Hansova for Professor Huber. We have seen several patients suffering from severe proteinuria, sometimes nephrotic without collapsing GN focal segmental glomerulosclerosis. It looks like acute kidney injury and podocytopathy. Do you have similar experience?
Yes, so we very often, so in most cases, we see a very slight more tubular proteinuria, which could be due to direct damage of the virus or due to the circulating cytokines and hemodynamic factors. Larger amounts of proteinuria we do see, but this is much more rare. And more commonly, this was my impression, seen in the United States and among African Americans. Some of them might have an upper L1 variation. This is, and many of these cases then were being associated with collapsing FSGS. But don't forget, we have so many patients now with COVID-19. We know that there's also a virus-associated minimal change disease. Many things that we see in other infectious diseases could also, of course, be seen in COVID-19.
Another question for Professor Huber. Yes, from Jolanta Kowalewska. Does underlying glomerular disease, glomerulonephritis, diabetes nephropathy increases the risk for COVID-associated nephropathy? And I would add also, does the situation in patients after kidney transplant, is this another risk factor for COVID-associated nephropathy?
We don't know even how we exactly define COVID-associated nephropathy. We are very aware that, general risk factors for severe courses of COVID-19 are chronic kidney diseases, diabetes, so the things that just have been mentioned are per se risk factors for severe causes of COVID disease. And we know that if people are severely sick and going to intensive care unit or using mechanical ventilation, then they have a very high risk, up to 30 or 33% of also developing acute kidney injury. So yes, chronic kidney diseases itself, diabetes, and factors leading to chronic kidney disease are per se risk factors of severe causes of disease and thereby also of an involvement of kidneys. Whether I would specifically call this COVID-19 nephropathy, I would not be sure.
Okay, thank you very much.
May I have a question to Dr. Huber, or it's just reserved only for the participants from outside? Now, my question is, what is your opinion about the Harald Miszak is now doing the proteomics in the urine. Do you think that we can obtain something more information after this proteomic analysis of the urine, or is just a scientific idea behind?
No, I mean, we always can learn by more deeper and omics-based, unbiased methods. So what they're doing here is is rather to develop a panel that predicts chronic kidney disease progression. So it involves certain stress markers and proteins, and that certainly will also be affected in COVID-19 associated tubular injury and damage. And people observed, for example, that markers like DICOP3, one of the more recent markers for tubular stress, it might also be increased in these diseases. So I think it's very worth looking for this. Yes.
1/3 of the patients progress to the chronic kidney disease. These are the data from the literature that 1/3 of these patients after acute kidney injury will progress to the chronic kidney disease, which is not very benign condition.
No, and I think this is important to make from the viewpoint of a nephrologist, that nephrologists need to be involved in the intensive care unit and taking care of these patients, because it has been very commonly overseen that acute kidney injury is not only an acute disease, but is commonly followed by an increased rate of CKD, and these patients need to be very well supervised. And we have now new medications at hand to slow down progression of chronic kidney disease. With that, it's even becoming more important.
Thank you so much. Due to time constraints, we have to stop at this point the question and answer part and move to the second part, to the further part of our meeting. And all questions that remained unanswered will be addressed by the lecturers later on via the chat box. And before we take a short break, I would like to remind you that all the lectures will be available for you after the conference as a video on demand on the IRPC devoted platform till the end of the May 2021. And thank you so much, the lecturers and the moderators of the first session. It was fascinating, and we shall see you at 1:40.
Good morning to all the participants. My name is Michele Rossini. I am a nephrologist and a renal pathologist at the University of Bari, Italy. I work with Professor Loreto Gesualdo, and I'm giving this talk instead of him since he's fully devoted during this day
Okay, let us move to the next part of the session. Professor Durlik. Professor Durlik, Professor Rocco. Can you?
Ladies and gentlemen, it is my pleasure to introduce the next speaker, Professor Michele Rossini from Italy. Professor Rossini is head of renal pathology department in the nephrology unit in the University of Bari, and he will tell us about Omics in kidney diseases, tissue proteomics in contemporary nephrology. Professor Rossini, the floor is yours.
Good morning to all the participants. My name is Michele Rossini. I am a nephrologist and a renal pathologist at the University of Bari, Italy. I work with Professor Loreto Gesualdo, and I'm giving this talk instead of him since he's fully devoted during this day to face the COVID-19 pandemic situation. So, tissue proteomics in contemporary nephrology. The traditional approach to renal biopsy analysis is to identify the pathology by systematically examining the different histologic compartments of the kidney, glomeruli, tubules, interstitium, and blood vessels. Once the site and the nature of the lesion are determined, the pathologist makes a final diagnosis integrating findings from light microscopy, immunofluorescence, electron microscopy studies, and clinical history. This approach has been challenged by the current era of big data in basic and translational nephrology research. The rapidly expanding collection of data on patient phenotypes and molecular tissue composition that is being generated through omic technologies can lead to molecular phenotyping of diseased tissue, and offers an opportunity to advance patient care by improving diagnostic precision and mechanistic understanding beyond that achieved through conventional histological approach. The integration of multi-level data from genomics, transcriptomics, proteomics, and metabolomics play a crucial role in determining the molecular changes related to disease progression and early pathological glomerular modifications, drive the identification of potential molecular subtypes of the disease, and may contribute to the development of new non-invasive biomarkers for a better molecular characterization of renal damage and its follow-up. According to the central dogma of molecular biology, the proteome is encoded by the transcriptome. So we have 20,000 protein coding genes that encode more than 200,000 transcripts that give rise to the proteome, which probably includes over 2 million proteoforms. The activity of a protein not only depends on its level of expression and gene-encoded primary structure, but also on a plethora of regulatory biological processes, including post-translational modifications giving rise to different proteome layers. So we will have the ubiquitinome, the acetylome, the phosphoproteome, the interactome, etc. Proteomics is becoming a highly useful tool in nephropathology, providing an insight into the molecular aspect of the disease and aiding in the identification of candidate biomarkers for both diagnostic and prognostic purposes. Many proteomic approaches have been applied in the study of glomerulonephritis, analyzing urine, serum, plasma, tissue, following an almost common flow chart. First, you have sample preparation with prefractionation in order to reduce sample complexity. Then you have the analytical part with protein-peptide separation that can use different techniques. You can use two-dimensional gel electrophoresis. You can use, like in this case, in this picture, MALDI mass spectrometry or liquid chromatography mass spectrometry. And then you have the analytical part with data elaboration, with protein identification, and finally pathway analysis. Biomarker discovery has focused on easily accessible analytes such as serum, plasma, urine, but the identification of novel biomarkers in these biological specimens has been sometimes disappointing. Failure to identify biomarkers with clinical impact results from the complexity of identifying low abundance protein in fluid dominated by high background concentration of proteins, such as albumin in the urine. Tissue proteomics is based on the premise that biomarker discovery will be facilitated by analysis of the primary site of pathology, in this case the glomerulus. Once novel molecular markers have been identified and validated with the use of a tissue sample, more targeted analytical approaches to identify low abundance molecules can be applied to easily accessible samples such as urine or plasma. An example of this is the identification of phospholipase A2 receptor as target antigen in idiopathic membranous nephropathy. In this paper, the authors isolated normal glomeruli from kidneys that were unsuitable for transplantation. Then they conducted a Western blotting analysis of protein extracts from normal human glomeruli with sera samples from patients with idiopathic membranous or secondary membranous or other proteinuric or autoimmune disease. They identify protein bands that reacted with the antisera from idiopathic membranous patients. These antigen bands were then excised and analyzed by means of mass spectrometry. And mass spectrometry generated a working list of candidate proteins. Then the authors used available antibodies and recombinant proteins to test candidate antigens and found that antibodies against phospholipase A2 receptor identified a glomerular protein band of the same size as that recognized by the serum from a patient with membranous nephropathy. The same study design was used in this other study that led to the identification of another antigen in idiopathic membranous nephropathy, the thrombospondin type 1 domain containing 7A. A proteomic approach was also used to identify antigen involved in fibrillary glomerulonephritis. In this case, glomeruli from fibrillary glomerulonephritis patients and controls were isolated from formalin-fixed and paraffin-embedded biopsy specimens using laser capture microdissection. The controls in this case were normal kidney plus other disease that included diabetic glomerulosclerosis, different type of amyloidosis, light chain deposition disease, and idiopathic nodular glomerulosclerosis. Proteins extracted from these glomeruli were analyzed with liquid chromatography and mass spectrometry. These studies identified DNA-J homolog subfamily B member 9 as a highly sampled protein detected only in fibrillary glomerulonephritis cases. Then immunofluorescence and immunohistochemistry studies with an anti-DNA-JB9 antibody showed strong and specific staining of the glomerular tufts in a distribution that parallels that of the immune deposits. In this study, mass spectrometry was done on protein extracted from glomeruli, microdissected and isolated from biopsy specimens of phospholipase A2 receptor negative membranous nephropathy and from phospholipase A2 receptor positive membranous nephropathy and control case, and led to identify other two candidate antigen, exostosin 1 and 2, as a target antigen of membranous nephropathy. Proteomic analysis has also the potential to better classify kidney disease. An example of this is the MPGN pattern of disease. Historically, membranoproliferative glomerulonephritis were classified according to the ultrastructural aspect of the deposits in MPGN type 1, type 2 or dense deposit disease, and type 3. More recently, a new classification has been adopted that takes into account immunofluorescence findings that more accurately reflect pathogenetic mechanism. In this new classification, the term C3 glomerulopathy has been introduced and includes dense deposit disease and C3 glomerulonephritis, where immunofluorescence identify only C3 with no or little immunoglobulin. The group from the Mayo Clinic conducted proteomic studies on glomeruli that were microdissected and isolated from kidney biopsy specimens of patients with dense deposit disease and C3 glomerulonephritis. And they found that based on proteomic studies, it appears that the complement profile in both C3 glomerulonephritis and dense deposit disease is similar, thus confirming the pathologic classification. Furthermore, they also noticed in these proteomic studies that there was no factor B present, indicating absence of C3 and C5 convertase in the glomeruli, suggesting that activation of the alternative and terminal pathways occurs in the fluid phase rather than resulting from local disturbance of the alternative pathway in both C3 glomerulonephritis and dense deposit disease. Mass spectrometry can also be used to obtain molecular images of tissue. Mass spectrometry imaging is a technique used to visualize the spatial distribution of molecules by their molecular masses. In a typical multi-imaging mass spectrometry experiment, a thin tissue section is mounted on a microscope slides and then coated with a multi-matrix. This matrix is typically a small organic acid with a strong absorbance at the wavelength of the incident multi-laser. Then the laser desorbs and ionizes analytes mixed with the matrix molecules, generating a mass spectrum at each XY coordinate, and this generates a raster images of the tissue surface. So, using this acquired mass spectra, the spatial distribution of any of the biomolecules present can be visualized and a molecular image of the tissue can be reconstructed. And here is an example where on the left you can see the PIS of the kidney, and on the right the corresponding molecular images obtained with multi-imaging mass spectrometry. Multi-imaging mass spectrometry is a rapidly developing technique that now allows the simultaneous analysis of the distributions of up to hundreds of peptides and proteins directly from a tissue section. This technique uses the masses of peptides and proteins to distinguish between different kinds of proteins and thus does not require any form of labeling. These profiles can be used to obtain biomolecular signatures which correlate with the specific histological features. and this has been used to distinguish between different regions within a tissue and to differentiate and classify tissues. And this is why it has been initially used in the oncologic field. In renal pathology, amyloidosis is a candidate disease to evaluate by multi-imaging mass spectrometry. The diagnosis of amyloidosis relies on the identification of characteristic green birefringence in polarized light after staining with Congo red. To date, more than 30 different peptides and proteins are known to form amyloid fibrils, and since the patient's prognosis and treatment depend on the amyloid type, an accurate diagnosis and classification is of great importance. So far, after identification of amyloid deposits by light microscopy, immunostainings and immunofluorescence studies are conducted in order to identify the amyloid protein. Recently, laser microdissection coupled with liquid chromatography and mass spectrometry was demonstrated to be a valuable tool for the typing of amyloidosis and is currently the most widely adopted proteomic strategy in the field of amyloid diagnostic. But a point to be underlined is that the above methods require the presence of amyloid deposits using the specific dye Congo red. This is due to the fact that proteomics provides information on the identity and sequence of protein in the tissue, not on their conformation. However, analysis of the proteome profiles of amyloid-affected samples showed that a set of proteins known to colocalize with amyloid deposits can be simultaneously identified by mass spectrometry in amyloid-laden tissues. This includes mainly apolipoprotein E, serum amyloid P, and vitronectin. The simultaneous mass spectrometry-based identification of multiple amyloid-associated proteins in a specimen indicates the presence of fibrils with high specificity and sensitivity, correlating well with Congo red staining and representing a kind of universal amyloid proteome signature, proposed as novel independent markers of amyloid deposits. In this study, we have an active example on how proteomics coupled with imaging tools can be used in renal pathology in the field of transplant pathology, to be more precise. In this paper, the Fourier transform infrared spectroscopy was used to quantify fibrosis and inflammation, which are two of the main histological components that induce the loss of a renal graft. And fibrosis and inflammation so far are routinely assessed by renal pathologists by visual examination of stained slides with a notable lack of reproducibility and accuracy. In this study, the infrared spectroscopy was used to build, without staining, biochemical spectral images with a spatial resolution of a few micrometers. And from infrared color-coded clustered images, a classification model was developed to automatically detect and quantify renal fibrosis and inflammation. In these other studies, multi-imaging mass spectroscopy was applied to formalin-fixed paraffin-embedded renal biopsies from histologically diagnosed primary and secondary membranous nephropathy patients in order to detect alteration in their tissue proteome. multi-imaging mass spectroscopy was able to generate a molecular signature of primary and secondary membranous nephropathy, with one particular signal being overexpressed in the glomeruli of primary membranous nephropathy patients respect to secondary membranous nephropathy ones. The highest relative intensity of this signal correlated with the glomeruli that were affected by the glomerulopathy in primary membranous nephropathy, while this increased level of intensity were not noted in the affected glomeruli in secondary membranous nephropathy patients. So in this case, multi-imaging mass spectroscopy was used basically to hide in differentiating primary versus secondary membranous nephropathy. So in conclusion, the generated proteomic data complements the omics data produced by other high-throughput technologies, with the main purpose to gain crucial insights into disease biology and pathology that could lead to the development of more complete predictive models for a better stratification of patients in order to provide them with targeted therapy and contributing further to the field of personalized medicine. Thank you for your attention.
Thank you very much, Professor Rossini, for your excellent lecture. Discussion will be held at the end of session. Please stay with us. Thank you.
Okay, so let's continue this molecular pathway of this session. Professor Krzysztof Mucha from the Department of Immunology, Transplantology and Internal Medicine at Medical University of Warsaw will present the data concerning urinary proteomics in kidney diseases. Please, Professor Mucha.
Everybody, it is a privilege to have a lecture during the International Renal Pathology Conference, and therefore I would like to thank the President of the Renal Pathology Society, the organizing committee, and of course special thanks to Professor Agnieszka Perkowska-Ptasińska, who made a huge effort to organize this conference. This how the urinalysis looked like for the last 100 of years. But thanks to the new technology based on omics, nowadays we can use the one sample for a genomic studies, for a metabolomic studies, and of course also for a proteomic studies, which is the topic of today's lecture. But the way to the proteomics we know today took us more than 100 years, and it started in 1899 when Thomson developed the first mass spectrometer. Then in 1959, the gas chromatography was used for a peptide sequencing. And in 1966, the next step was the use of tandem mass spectrometry. The term proteomics was proposed and first time used by Mark Wilkins by analogy to genomics, proteomics. We should also remember that 5 Nobel Prize winners worked in mass spectrometry. The first one, of course, was Thomson, who received a Nobel Prize in 1906, then Aston in 1922, Paul in 1989, and finally in 2002, Fenn and Tanaka. If we think about the proteomics studies in the urinary tract diseases, the first publication was released in 1997, and it was about the proteome profiling of bladder squamous cell carcinoma. So the study was done on the tissue. the first study, the first proteomics study done on the urine is from 2001, and the authors analyzed and presented the proteins profile of healthy human male urine. So the proteomics is relatively very young methodology that we are using for last 20, maybe 23 years in medicine, which is nothing comparing, for example, to the genomics. The proteomics technology is divided into 3 phases. The first, discovery phase, also named as shotgun proteomics, which is simply based on the testing couple of samples and try to find an interesting proteins for future researchers. Second phase is a verification, also named targeted proteomics, and the third one is a validation phase, primarily based on the immunoassays. But now, as the mass spectrometry (MS) is widely used in the diagnostics, then the validation phase is based on the mass spectrometry. Therefore, the last 20 years we can divide into the period before 2010 and after 2010. If we look into the PubMed and try to check how many publications regarding nephrology and urinary proteomics were published within the last 20 years, we found that there is a less than 500 publications. And of course, less about glomerulonephritis, because there is something between 160, maybe 200 of publication about glomerulonephritis and urine proteomics. And these 200 are divided into less than 30 about ADPKD, something about 40 about IgA nephropathy, less than 30 about FSGS, and something between 40 and 50 about lupus nephritis. Of course, it depends how you ask the question, either urinary or urine proteomics or proteomics alone, then the number of publications slightly increased. One of the first kidney diseases that was studied with the urine proteomics was ADPKD, and this is one of the publications from 2018. On the left-hand side, you see a list of 109 peptides representing 69 proteins. But what is much more interesting, this is on the right-hand side, the heat map of these unique peptides representing 69 proteins. The red color dots indicates upregulation of the protein, and the blue dots indicates downregulation. The column, this is the single protein, and the row, this is a single peptide. And thanks to such studies, we can try to find a panel of proteins that might be involved in the physiology and pathophysiology of the disease, namely in this case ADPKD. And as you see, this panel indicates the proteins that are downregulated in the patient with ADPKD comparing to the healthy individuals. This is another publication done by our group and published in 2012. And as you see, in 3 experiments, more than 1,000 proteins were common. Out of this, finally, there is a list of 155 specific for ADPKD proteins comparing to the healthy controls. And one of these proteins is or was retinol binding protein. And just have a look on this picture. On the right-hand side, you see patients with ADPKD, and on the left-hand side, you see expression of retinol binding protein in healthy control. The next one, glomerulonephritis. The next one, kidney disease, is IgA nephropathy glomerulonephritis, and this is one of the publications touching this subject from 2015, showing biomarkers discovered by urine proteome analysis. And as you see, there are some proteins that might be up or down-regulated in patients with IgA nephropathy. This is another publication comparing IgA nephropathy and diabetic nephropathy patients, and the authors finally discovered and listed 5 specific for IgA nephropathy proteins. It is interesting because these proteins are completely different from another publication, and the answer lies in the control group, which in this case was a diabetic nephropathy, not a healthy individuals. This is a publication from 2017 summarizing the research is done in IgA nephropathy and urine proteomics within the last 10 years. And the authors proposed a distribution, the physiological distribution of 68 shortlisted proteins based on the study done in last 10 years. As you see in the table, the last study was done by us, and we did it in the 2013. We compared the IgA nephropathy patients to healthy controls, and we found in 3 experiments more than 600 proteins common. What was very interesting, finally we defined 31 specific proteins for IgA nephropathy patients with 8 proteins that were never previously described in IgA nephropathy. Out of this, we were able to propose a linkage between 19 specific proteins, the physiological linkage, what was very interesting and very important from our point of view. In the next step, we decided, and we would like to verify what we have found in mass spectrometry. Therefore, we performed a Western blotting. And on the left-hand side, you see one of the blot with the glomerulonephritis patients on the left-hand side, on the red, and on the green, the healthy control. And we found both the whole protein band density were different between diseases, namely IgA membranous and lupus nephritis, as well the upper, middle, and the bottom range bands differs between diseases, as well between diseases and the healthy controls. And I would like just to show you shortly how it looked like. The control group looked like, as you see, then IgA nephropathy, the membranous nephropathy, and erythrocyturia of unknown origin. These groups of patients were quite similar one to another, but if we compare the lupus patient, some of them look like a control group, some of them look like a IgA nephropathy or membranous nephropathy, and another look like erythrocyturia. Based on our findings, we sent a patent application for methods of diagnosis, differentiation, and monitoring using urine proteins as a marker for IgA nephropathy. We are after review and waiting for a final decision. Where are we now? Remembering the picture I showed you before about 3 phases of proteomics, we didn't stop our efforts, and we increased the new identified urine proteins, and now we have more than 2,600 proteins. And these are the data from November to 2020, comparing with 1,238 proteins into 2014, which means that we have now twice more proteins defined and identifying than 6 years ago. And our network the physiological interaction networks between proteins in 2014 looked like you see on the left-hand side, and now we have more proteins, more very tight linkage between all those proteins, which is very important, and it proves that these proteins are very important in the pathophysiology of glomerulonephritis we are studying. Having all this data, all the information about proteins found in glomerulonephritis, there are two possible ways of thinking and working. One is try to find this holy grail protein, the one and only responsible for physiology and pathophysiology of the certain disease. Another way is try to define a panel of proteins that might be responsible for the same. However, we are looking for this holy grail protein, but firstly, we try to define a panel. And this is the picture from our patent and publication. That's why before publishing it in the spring to 2021, we cannot show you the proper name of proteins, and that's why they are covered, the protein A, B, C, D, and F. And the colors are the different types of disease. The green is disease 1, red disease 2, the blue one, disease 3. If somebody would like to diagnose the disease number 1, then the protein A and B should be checked. If somebody would like to diagnose or monitor the disease number 3, then the protein C and D should be checked. And this was a fundament for our screening proposal tests that we just sent a couple of days ago. We defined five proteins, including albumin, and again, sorry that we cover this name of the proteins. And based on the mathematical algorithm, we are able to exclude the patients that are suspected for other diseases than IgA membranous or lupus nephritis. The accuracy of this test is almost 98%, and this was sent to as a patent application on November 10th. The second stage is how to differentiate this patient suspected for IgA membranous or lupus nephritis. And this is, as we call the proteins tree. There are proteins that were defined based on the proteomic analysis. And for example, if we have the protein 1 on the concentration above 8.4 and the protein 2 and the protein 3, then the patient probably has the lupus nephritis. If we have the patient with the protein 1 above the concentration 8.4 and protein 2 less than 4.0, then the patient probably has IgA nephropathy. And if we have another proteins tree, then the patient has probably membranous nephropathy. Probably means that the probability is something between 70 till 80%, which is not bad. There are still some missing answers in the urine proteomics. First of all, why we are using MS? The answer lies in the power calculation. If we would like to have a proper power calculation on the level 0.9, and we use ELISA, we should have more than 1,900 patients, which is a lot, comparing to less than 600 with the use of MS. Secondly, there are many publications with different sampling. Namely, in one publication, you can find samples from 24 hours urine collection. In another, you find samples from after the night, and another a spot urine. So this must be defined and clearly said which methodology is the best. The third question, have we lost the proteins during our urine proteomics? Of course, yes, because the cutoff is on the level of 10 kilodaltons, then we have an absorption phase and degradation. So the answer for this question, have we lost, is yes, we were lost the proteins. But the question, much more important, are they very important or not? Nobody knows. The fourth, my favorite, 24 years ago appeared a paper in New England Journal of Medicine showing that there is a probably circulating permeability factors. And even though that for last 23 years we're using MS in diagnostic and medicine, nobody knows the name of this factor. Finishing the lecture, once more, I would like to thank you for the invitation. I would like to thank you for all the authors of our job. The name of the authors are here. The friends from my department, Professor Agnieszka Perkowska, whom you know very well, our friends from Polish Academy of Science, and a special thanks to Dominik Cyseski, and our friend Professor Krzysztof Mucha, well known from genomic studies, but he had also a great input into our work. This how the urinalysis looked like for last 100 of years, and I hope that in next years the urinalysis will be based on the proteomics studies. Once more, thank you very much for your attention.
Thank you very much, Professor Mucha, for this very interesting presentation. Both papers are now open for discussion.
May I have a question to Professor Mucha? Professor Mucha, I would like to ask you, can you hear me? Yes? I think you can hear me, yes?
And do you hear me?
Can you hear me?
Yes, yes, I hear you. And you?
Yes, of course. I would like you to ask you if this pattern of proteins in glomerular diseases depends on CKD stage. For example, it's different in CKD stage 2 and 3 and 4. So my question is if it is possible not only to diagnose but also to monitor the progression of glomerulonephritis, response to therapy, and would you comment?
Yes, this is of course the crucial question, and doing our study we try to match the patients sex to sex and how old the patients were, of course the CKD and the level of the proteinuria. But this part of the study was a study we involved less than 100 patients. The next part, based on our experience together, what we got from Professor Perkowska, because one missing slide was that, of course, Omics is not the holy grail without the histopathology. And this is the brilliant platform for cooperation between the clinician, the basic scientists, like our friends from Polish Academy of Science, and the histopathologist like Agnieszka. So answering for your question, we should do a validation study, and we calculate this, and we need something like 700 patients from these three groups of patients, I mean IgA, MN, lupus nephritis, plus control group. And then we could answer, maybe not fully, but much more precisely than now. Till now, we took a patients, let's say, till the stage 3. We didn't took a patients with the GFR less than 15, because from our point of view, it was useless.
Thank you.
I have one question to Professor Rossini. Recent guidelines suggest that in patients in whom antibodies against phospholipase A2 can be detected, kidney biopsy is not necessary to perform. You showed us the data from mass spectrometry. that can also distinguish primary from secondary glomerulonephritis. Do you think it is also sufficient to recognize primary glomerulonephritis to not perform kidney biopsy in these patients? Your microphone is turned off.
Can you hear me?
Yes, now, yes, okay.
Okay, I was telling that as a nephrologist that also performs renal biopsy, if I have a patient with a positive anti-phospholipase 2 receptor, I would rather prefer to not doing the biopsy to avoid the risk of complication. But since there are patients that are positive for the phospholipase 2 receptor antibodies and also has some secondary disease, in this case, if there are not contraindications for renal biopsy, I rather prefer to perform renal biopsy, because not only tell me of course, confirming that it's useful to confirm diagnosis, but also to provide also prognosis in this case. The only probably condition where I'm not going to perform the biopsy is when the patient is a solitary kidney or a very advanced kidney disease where the risk is not worthy to to perform the biopsy. The balance between risk and benefit.
I am also the same opinion about this indication to biopsy. Thank you very much.
Dear moderators and speakers, thank you so much for this session. I would like to attract your attention to the fact that there is a reviewer section on the platform. We would appreciate your opinions and comments concerning the conference, and you are welcome to use the questionnaire that can be found there. Again, our distinguished lecturers and moderators, thank you very much. And all participants, ladies and gentlemen, we shall see you at the next session that starts 2:45. Thank you so much.
Thank you.
Raj.
Bye.